Bismillahirrahmanirrahim. I hope you all find in this fundamental condition. So, in this situation, I tell you about the study of the Drosophila. I mean to say how we study the genetic structure of the Drosophila fly, and what are the procedure for studying this fly. How we prepare the Drosophila fly. That it becomes easy to study about the genetic structures and its sexes and transmission, how traits transfer from parents to offspring. What are the procedures that's involved in transferring of the traits, and how we prepare the Drosophila fly for our studying the genetic structure. So, for these purposes, let's start our video lectures. That is about the preparing the Drosophila for studying a genetic structure. So material required for this purpose is Drosophila nonresters, and it is the scientific names of the Drosophila, that is the small fruit flies, and the microscope used for this purpose is stereo microscope. So the common fruit fly is basically the genetic structure. So that is the view of the microscopic view of the Drosophila fly, and who is the first scientist? That are Thomas and Morgans were the first scientists who used this the Drosophila for studying the genetic structure. That in 1909 it was first studied the genetic structure of the Drosophila. But before studying the Drosophila, he prepared the Drosophila for studying genetic structure in the following procedure, such as first of all, we tell you about the knowledge. A single Drosophila fly can lay about 500 eggs. Understand? 500 eggs can lay. So, mean to say, its reproductive stage of is too fast. Is too fast. So, for studying genetic structure, because it can reproduce too rapidly. So, mean to say, it can lay about 500 eggs. So, its genetic structure can study very easily. And its reproductions with the minimum time take place at a very fast speed. So. First of all, you know that about the phenotype, means its coloring. As you looking here, it's above about white eyes, and the other is about red eyes Drosophila. Mean to say they are easily to large enough to studying about their eye characteristics, and their body colors is easily to study when we look at the phenotypically when we look at this fly. So. First of all, and what are the purposes that the Morgan use this fly? The following characteristics, for example, this fly. The first, this fly can easily be cultured. Mean to say, it easily can reproduce at temperatures about 21st, and in generations time for 2 weeks. Mean to say, for 2 weeks it can be easily generated at the temperature of 21st centigrade. So mean to say, its reproduction state is too fast, as you know. So mean to say, it can lay about 500 eggs. So mean to say, it can reproduce too fast. So for that purposes, for the maggots use this for studying. So what are the chemicals that are used for culturing? That's the both are used for real. I mean, the real is basically the chemicals. So the chemicals both are used for storing the Drosophila is basically the vacuum plastic like bottles, transparent bottle, and they are considered as a culture vessel easily available in the market and other laboratories, institutions. And when we study about Drosophila, so then we prepare a culture, its storage of the Drosophila, and it maintain a specific temperature. So we also provide the specific type of food, that means 5 grain of the yeast, and they are totally mixed in a chemical that is known as various chemicals, and equal volume, and also the water equal volume. Both the yeast and the water are stored at equal volumes in a real bottles, that is not a plastic bottle, for providing to the Drosophila fly. So they are first in their life cycle of the Drosophila. That was resembles by during the studying the genetic structure of Drosophila. First of all, the male and females and the transfer of the gametes and forming zygote, and outside of the body they're the embryo form. So they're about 8 to 10 days, and basically the 8 days for the formations of the larva, and 6 days for the formation of the pupa. So the embryo forms, and egg to mature adult, mean to say that 10 days, 8 to 10 days are required for conversion to the eggs to mature adult, and the temperatures required for in Fahrenheit that it is 5. At this temperature, the egg hatches, means eggs burst in about 30 hours. In 30 hours, the egg burst, and the larva feed for 5 to 6 days, and then pupate. So it means that 8 days required for formations of the embryo, and the 6 days required for the formation of pupa. At temperatures is that maintains at 84 Fahrenheit. So at these temperatures, the egg bursts and hatches about 30 so by continuous maintaining temperature that is 85, and egg burst, and embryos and come out, and converted into the pupa, and as a result with the passage of time is transferred into form between the days after, after 14th day, maximum 14th day, it's converted into mature adult, and the mature adult is ready for rapid continuous reproductions. So the basic stages, first of all the embryo and one install larva, and the second install larva, and then third. There are 3 stages of the larva, and first, second, and third, and that is converted pupa. Pupa means that the before stages of the pupa, before becoming the pupa, and after this stage, first when the 3 stages complete, and the pre-pupa form. So in the next stage it will be ready to form the pupa and then converted into female adult. Understand? So this is the basic life cycle of the Drosophila. So 2 days after emerging, a female start laying eggs. Understand? So you know that there are 4 basic stages, and the embryo first, install larva, and second install larva, and third. So it continuously, when we maintain the temperature 35 Fahrenheit, it directly convert egg hatches, and embryo converted into larva converted into pupa, and directly converted into mature adult. So the mature adult, if the females, then it become ready to lay eggs. You know that the Drosophila melanogaster fly, that's a small fruit fly, lay about 500 eggs at a time. So it was extremely important that adults remove from culture when you study, and you know that reproduction stages move forward from the females. So that's why when you remove adults fly, when the females and when the flies become mature, then remove all adult flies from 8 to 12 over. Before it is used for the selection of the virgin female. So in order to detect that the female, unmarried female, mean to say virgin female means, so before sections of this female characteristics of females, so remove all adults fly from culture 8 to 12. Mean to say, if the female is converted into mature adults, if embryos, larva converted, and then pupa, and then adult mature fly. So after this sessions, after this stage, for example, remove all the adults fly. Understand? Because at this stage it becomes easy to study about the female characteristics of the Drosophila. So that's why it's removed from the cultures. Because if we mix it, and the female can lay eggs, and gametes form, and again reproduction start, and we unable to visualize the females and the male, and unable to study about the genetic structure, that genetic traits of the Drosophila, and how it's transferred from the parents to offspring. So for this purpose, when you remove this fly, and you fly naps basically the chemical that is used to anesthetize the fruit fly. So this is basically chemical, understand? Then you anesthetize the fruit flies. So the later cultures and the beers, and while anesthetizing, so that's why that they later cultures beers. Mean to say the cultures chemical, the chemical both that contain the cultures of the Drosophila flies, for example, puts them aside, and now why mean to say the fly move from the culture box while anesthetizing the fruit fly. So dip the wine, mean to say take a stick, little stick, and that is used to enter the in the culture box, for example, and take a small dip into the fly naps beer solution, and rub up the wand, and rub up the wand, and remove extra chemicals from the end of the wand, and insert it into the culture box that contain the, mean to say, insert it into that on this box, in this culture box, which contains the Drosophila flies cultures, as well as Drosophila cultures. First, no cultures culture that contain the Drosophila cultures, and dip this wand that contain the chemical traces and places on those cultures vessels that contain the Drosophila. As a result, the flies become anesthetized in a state of unconscious stage, and at the stage, for the first, stick the anesthetic strip, mean to say tips of the wand into the beer just before the plug. Just before the opening of the vessel, and you dip this wand stick that contain anesthetics events that are used for the anesthetize the fly. Just dip this wand into the vessel. Understand? Just dip, because if you dip, maybe it go into the death state. So I just need to anesthetize the fly. Slightly amount of the chemical inserted into the vessel that contain the cultures of the culture of the Drosophila, and it's a fruit, and that contain the 5 grams of the yeast and plus water in equal volume of amount. So when you just this, you notice that our fly are anesthetized. Understand? Our fly are anesthetized. So now immediately lay the flies on the paper, and you have 50 minutes. But when after the anesthetize the fly, when the fly go into the unconscious state, you only have the 50 minutes to studying it. So place the paper on flies and move them with a fine brush with separate different kinds and sexes, and discarded fly, meaning the fly which have gone into that state, and place in the morgue. I mean to say the rooms that contain the death's bodies, and similarly you remove this fly into the morgue. So divide the fly by the type and sexes, mean to say that white fly crosses with the white as fly crosses with the red as fly, and similarly you cross this fly. So this is the markers that contain the fly, dead fly. So male fly have dark and abdomen, and the abdomen is round. Means you notice that the male, when you study the Drosophila, when you notice the characteristics of the rounded abdomen and the rounded abdomen and this dark, which is dark at the end. So you notice that it's a male fly, Drosophila fly, and when you looks at the female, it is pointed at the abdomen side and no dark in color, and it is a female characteristic of Drosophila. So here's that is the no sex comb, and sex comb is also the other characteristic or determinations of the Drosophila. When you notice that the sex comb places over the arm of the Drosophila, then it is the female. Male. And when there's no sex combs presence at the pointed end of the Drosophila, then it is the females, and it usually has the pointed long abdomen, and this is not dark in color at the ends of the abdomen, while the male it is mostly dark at the end of the abdomen. So you look again here, that is the basic characteristic for determination of male and differentiating the male and the female. So the matings and the counting, we are labels with the characteristics. So the parents 6 guardians and 6 where females are present in the culture we understand. You must note it. The first of all, you take bottles and takes fruit fly and places over here in the vessels that containing the chemical, that containing the cultures, and that 5 gram of the yeast and plus equal volume of the water. And when the fly at the reproductive state, you know that the reproductive state of the fly is too fast, it can lay about 500 at a time, and you notice while looking at this, and you can notice about the reproductive life cycles of the Drosophila by keeping at temperature of 85 Fahrenheit, and after 14th day, mean to say 8 days for larva and 6 days for pupa developments, understand. So after this periods, you notice that it can directly convert it into the mature adult. So remove mature adults after the 14 to 15 over. So that is the basics for the identifications of the females, virgin female, understand. So 7-8 days after the process start, and remove the parents from culture and note about the reproductive life cycle. So that's about the end of the lectures, and in this lecture we study about, and after all this step, you notice the 7 to 10th day are able to studying this. We are need to studying this culture. So these are about the studying of the Drosophila fly, and I hope you understand. So if you like this video, please subscribe my channel and get the latest version of my video. Allah Hafiz from Swera.